Hello, this is Dr. Michael Edwards, founder and CEO of Bioinfo Solutions, a data analytics company based out of Parker, Colorado. This entry is part 3 in a series of instructional videos detailing a meta-analysis on 8 different human gene expression studies looking at papillary renal cell carcinoma. In this installment, I describe our downloaded papillary kidney cancer dataset and discuss formatting these data to combine into one analysis. I also demonstrate how we can upload the dataset into the statistical graphical platform Partex Genomics Suite for further visualization and analysis. A link to the actual Excel file I use in this meta-analysis can be found at a Google Drive link in the description of the video. So in part 2 of our series, what we used was Correlation Engine to put together a meta-analysis containing comparisons of our disease of interest, which in this case is papillary renal cell carcinoma. Given below are the genes that were differentially expressed in at least one of those comparisons. Again, the criteria for this is that the gene had to have a p-value of less than 0.05 and a absolute fold change greater than 1.2. So what we're going to do now is export all these results. And what we will export is not only the p-values for all these comparisons and the fold change, but what is really going to help our meta-analysis is we're getting the Illumina scores. So this is, again, as I mentioned before, a linear score based on from 100 to 0. So the highest fold changes in either direction will be 100, while the lowest fold changes will be a 0. So it's a very convenient way to get everything kind of on the same scale. Again, this is a linear scale from 0 to 100, 100 being the most highly absolute highest expressed fold changes and 0 the lowest. So we're going to take this, and all you have to do is go up here and hit export, and it will export all of this information into an Excel file. I'm not going to do that right now because it takes a little bit of time. So I've already have my Excel spreadsheet up, and I will show you what that looks like right now. This is the type of Excel file that will be exported from our meta-analysis. This top line here is basically telling you when you downloaded the data from Correlation Engine. These next series of rows are basically showing you the comparisons used in our meta-analysis. Again, this was focused on papillary renal cell carcinoma. And then this last line is the bookmark for this meta-analysis. So if you ever wanted to recreate it, you can basically copy and paste this to your Correlation Engine account, and it'll bring this up. My Correlation Engine account is different than yours, so what you would need to do is type in here your domain name. You would just basically put in the domain that you're using for your Correlation Engine account. And again, this will recreate the meta-analysis. Given down below are some of the genes that came up in our meta-analysis. I'm just going to use this first one as an example. It's giving you the gene symbol, the entries gene ID, so the number that identifies that gene, the full name for that gene, and how many of our studies that we looked at it was differentially expressed. So this particular gene was differentially expressed in 17 out of 17 of our studies based on that previous criteria that I told you about. This is the overall gene score. So again, I mentioned that Illumina is converting these fold changes, these absolute fold changes, into a linear scale from 0 to 100. So the greatest overall gene score we could possibly get would be the highest fold change in that particular study, which would be a 100. So if you added up all those 17 studies, the highest score we could possibly get would be 1700. And so we can basically rank them based on their overall gene scores. Here would be that rank score for bioset number 1, which is given up here. The p-value for that comparison, and then in this case the actual fold change. But again, we are using this ranking rather than this fold change for a meta-analysis. So what we want to do, but as you'll notice, is that some of these fold changes are negative, but again, remember this is based on the absolute fold change, so we have a positive score. So what we now would like to do is basically turn these score biosets, these Illumina scores, into positive and negative based on the relative fold change, the direction of that fold change. So that's exactly what we do on the next tab, is basically get rid of all this information, keep the overall scores for each comparison, and then convert them into either positive or negative values based on the direction of the fold change to that control for that comparison. So this is exactly, we'll do that on the next tab. So given on this tab are the normalized Illumina scores. Again, we took out all the fold change and p-value information, and again, we are now left with a scale from 100 to negative 100. So again, if that fold change in that particular comparison went down compared to the control, we made the Illumina score negative. Um, you can do this pretty easy. You can actually go back to this tab and sort by the p-value, say descending, and then just multiply all the score biosets by, say, a -1. I did not actually compile this. This was done for me by one of my good friends, Jim Flynn from Illumina. He's based out of Boston. He's an amazing bioinformatician, and I couldn't have done this without him. He's one of the few Patriots fans that I actually like. So, joking, that is a joke. The Patriots are a good team because you cheat a little bit. So, anyway, thank you very much, Jim Flynn, for putting this together. You can write code to do this, but by the time you sorted it and basically multiply by a -1, you're probably there. So again, this is the data set we're going to use. So this very next tab is basically the same data set. Now we've got all the headers. And this is what I'm actually going to paste into Partek's Genomic Suite. Again, we have our genes in this first column. We have how many data sets it was differentially expressed in, overall gene scores, and then again, the normalized scores for that gene in all of the different comparisons we used in our meta-analysis. I also want to say that this first tab, Summary, will give you more information. We're also going to download this into Partek, which gives us basically where the data set came from, the geo number where it was deposited, the study name, the comparison we actually used, the number of test samples, control samples in that particular comparison, the amount of genes that were differentially expressed in that comparison, and then different aspects of both the control and the test. What subtype or type of papillary renal cell carcinoma was it? In the control tissue, were we looking at, say, cancer versus cancer, or normal kidney tissue, or normal adjacent kidney tissue to the tumor? So what we're going to do is go back to here, and we are going to export this data into Partek and look at it even further.